Buongiorno e benvenuti alla nostra annuale giornata di aggiornamento. Bene, allora è mio grandissimo piacere introdurre il primo speaker della giornata, professor Lipuma. Se andate sul suo sito trovate pediatrician, ma è un pediatrician un po' particolare perché si occupa di microbiologia e in particolare delle infezioni batteriche. E come sapete ci interessano molto perché i batteri sono i principali nemici e quindi combattere i batteri è importante. Volendo guardare qualcosa di più su John, well, first of all is from Licata, originally the family, la famiglia, quindi ha del sangue italiano nelle vene. Se uno va su Google Scholar scopre una cosa interessante, cioè che ha un indicatore che si chiama H-index che ha valori molto molto elevati, 66, e questo vuol dire sostanzialmente che i suoi articoli sono molti e sono molto letti, sono letti da molte persone. È una delle persone che ha grande impatto sulla comunità scientifica internazionale. Sono 30 anni che si occupa di ricerca biomedica finalizzata alle infezioni batteriche, che vuol dire identificare il genotipo dei batteri. Voi sapete che esiste il genotipo degli humans, il nostro, e quindi sapete che esiste la caratterizzazione della mutazione della CFTR, ma capire come si comportano i batteri è un altro diciamo, complesso problema, ed è il problema di cui verremo a conoscerlo oggi attraverso la sua presentazione. Ieri abbiamo fatto un workshop con la dottoressa Lindsay Caverly, che è qui, che ci ha raccontato le loro ricerche in un gruppo di scientist. E si occupa di Burkholderia, Pseudomonas, Haemophilus, Pseudomonas, Acinetobacter, and so on. So tutti i tipi di batteri cattivi che in qualche modo avete visto nel corso della storia clinica della fibrosi cistica. C'è tanto da capire e su questa base I'm glad to invite Professor John Lipuma to give us his lecture about deepening, nuovi approfondimenti sulle infezioni delle vie aeree nella fibrosi cistica. John.
Grazie. Grazie mille, Professor Jerry. I trust you said some nice things about me.
Yes.
Grazie. È un piacere essere con te oggi. And welcome to those of you who are joining us remotely. Ciao Paolo. Come puoi dire, il mio italiano non è molto buono, quindi toro permesso parlare in inglese. Sì? Sì. Sarà meno doloroso per tutti noi. Bene. So there's a brief outline of the talk. We give a very brief history of microbiology in cystic fibrosis, very brief. And then we'll talk about some of the new approaches that we are using to understand infection in CF. I'll describe some of the early lessons that we've learned from this new approach. And then finally we'll end with some of the questions that are remaining that we are currently studying and trying to understand. So this is a starting point in some ways. Of course, cystic fibrosis has been with us for centuries, but it wasn't really until the 1930s, 1938, when Dorothy Anderson, who was a pathologist in New York City at Columbia University, first described cystic fibrosis. And she referred to this as cystic fibrosis of the pancreas. This was primarily described as a digestive disease where children with cystic fibrosis failed to thrive, and most children died at a very young age. Several years later, individuals like this person, Sidney Farber, started to understand the connection between cystic fibrosis and lung disease. And he made the observation that in cystic fibrosis, in addition to the digestive problems, children had obstruction of the airways, and he noticed that many of them had infection with Staphylococcus. Here's a table from a paper that was published in 1943, and what is described here are the findings from lung cultures of children who had died of cystic fibrosis. And you can see the ages of the children in months. Okay, most of them died very young. The oldest one is about 40 months old. And almost all of them had Staph aureus, in their lungs when they died. And so this really started to make this strong connection between lung infection and the problems that people with CF, children with CF, were having. So we have Dorothy Anderson in 1938, Sidney Farber in 1943, and around the same time that Anderson and Farber were describing cystic fibrosis, we had the development of antibiotics. So we remember that the sulfa drugs were really the first antibiotics developed in 1935. Penicillin is the one we think most about. It was developed in 1942, and then streptomycin in 1944. So you can see that the very beginning of the era of developing antibiotics coincided with the early descriptions of cystic fibrosis. These things happened in this very same period of time. Well, shortly thereafter, a number of other antibiotics were developed, and you can see them listed here. So a very rapid pace of development of antibiotics. These were very new agents. People weren't entirely sure how to use them, when to use them. And people came along, like this person, who's a well-known figure in cystic fibrosis, Harry Schwachman, who in 1960 proposed that we should be very aggressive about using antibiotics in cystic fibrosis. And he proposed we should use all of these antibiotics and use them early, use them aggressively, use them in different combinations, use them both by IV route and by inhaled. And people started to do this. They listened to Harry Schwachman and said, well, that sounds like a good idea. Maybe we should try this and see what happens. Well, what happened was quite dramatic. And you can see here on this graph, if we look at the different periods of time, so from 1952 to 1957, this shows the age of survival. And you can see that in that first period, 1952 to 1957, only about 30% of children with CF lived to the age of 2 years. Then the next 5-year period, 1957 to 1962, when these antibiotics were starting to be used, now suddenly the survival age dramatically increases. And by the period of 1962 to 1967, when Harry Schwachman and others were saying use a lot of antibiotics in CF, now look, over 80%, 90% of children are alive at 2 years of age, and 80% are alive at 5 years of age, versus about 10, 20, 30%, 15 years earlier. So very dramatic increase in age, and it's not, don't want to say that it was all due to antibiotics, because people were also paying more attention to digestive problems as well, but I would argue that a large part of this increase in survival was due to the aggressive use of antibiotics. Again, confirming the connection between infection and problems in CF. So it really made the early arguments about infection being important very clear treat the infections, children do better. Well, in the 20 years from 1960 to 1980, we sometimes refer to this as the antibiotic era. Many, many new antibiotics were developed during this time. As you probably know, the pace of development of new antibiotics has slowed considerably since then. Some people refer to this as the golden age of antibiotic development. Many, many new agents came on board, enabled us to treat many, many more different types of infection, different types of bacteria. And again, we see an increase. So in this 15-year period, 1965 to 1980, that coincided with the development of all these antibiotics, we saw a doubling of the survival age in the US. So the average survival, predicted survival age in 1960 was about 12 years, 11 years. And in 15 years, it doubled to 22 years of age. Again, coinciding with the aggressive use of antibiotics. Today, of course, we know that predicted survival is greater than 40 years. So we've continued to push that age up, and there's every expectation that that will continue to go up and up and up. Now, an interesting thing happened, though. With increasing survival and increasing age, the infection changed. The infection, as I mentioned, used to be primarily due to Staphylococcus bacteria. Well, now, as children became older, Pseudomonas aeruginosa, became the primary bacteria that we see in CF. Okay, so children aged, the staph sometimes remained, but as children got into their teens, Pseudomonas becomes the dominant bacteria. And this then resulted in what I call the Pseudomonas era in cystic fibrosis. And you can see here this timeline from 1970 to up until fairly recently, 2010. And what I've done here is I've put arrows pointing to the years in which very large studies were done that helped us understand more about Pseudomonas infection in CF. We understood about the epidemiology, in other words, the incidence, the prevalence of disease, how often it occurs, how bacteria may be spread. And the point I want to make here is that this is fairly recent. Okay? We sometimes have the sense that we've known all of these things for decades. In reality, it wasn't until the 1990s that we really started to understand more about Pseudomonas infection in CF. So this is not ancient history. This is fairly recent. Here's another timeline. All of these arrows point to the times when studies were done of antibiotic therapy for cystic fibrosis that was really targeting Pseudomonas. So there was a time when, again, as Pseudomonas became the dominant bacteria in cystic fibrosis, many studies were done to specifically treat the Pseudomonas infection because during this Pseudomonas era, this period of time, that was the major focus. Pseudomonas, the sense being if we could cure Pseudomonas infection in CF, we would be very far ahead. We would essentially be treating infection in general. So it's very focused on one bacteria. Now, the conventional approach to studying microbiology, infection in CF, is to use a standard culture. We culture the bacteria from the sputum specimens that the patients provide. This technology goes back 100 years. And the way it works is that we place some of the specimen, usually the sputum, sometimes the swab of the throat, onto a plate as growth medium, and we see which bacteria grow. Now, typically, the way we train the microbiologist in the laboratory is to see what grows on the plate and then pick certain bacteria that we believe are important in CF, because it turns out that oftentimes many things could appear on that culture, but not all of them are believed to be relevant. So we train the technicians in the lab, pick the things that look to be most important, that look like Pseudomonas, maybe look like staph. The other bacteria on the plate that grow, we ignore. And we tell the people in the lab, don't worry about those, because those are maybe not so relevant, maybe they're just from the mouth. And then, of course, there are many things that just don't grow on that media. It turns out that different bacteria require different types of media to grow. So if you just use one type of media, you're only going to get some of the bacteria to grow. You'll miss all the others. So this is what we call conventional approach. We can find more bacteria if we try hard, but it requires a lot of effort. So as I mentioned, some bacteria require special conditions to grow. Maybe, for example, no oxygen. Maybe some bacteria actually require the presence of another bacteria on the same plate to grow. Some bacteria cannot be grown at all in culture. They just don't grow. They grow in lung, but you can't get them to grow in the laboratory. So we assume that this conventional approach, the routine culture, has been missing many bacteria that may be present, either because they don't grow, or either because we ignore them because we don't think they're relevant. So a relatively new approach to studying infection, apart from CF as well, but we are now applying this to CF, is to use what we call culture-independent methods. So these are methods of detecting bacteria without the need to culture them in the laboratory. So instead of growing the bacteria, what we do in this case is we try to remove the DNA that belongs to the bacteria from the sputum, and then we analyze that DNA, and it tells us which bacteria are present, because each bacteria has a different code of DNA. So we can take the DNA, we can analyze the code, and it tells us what bacteria are there. We don't have to have it on the plate anymore. All we need is the DNA. This analysis allows us to profile the entire, what we call the bacterial community. So now we can see everything that's present in the sputum without relying on any growth in the lab. And an important piece of this approach is that it also allows us to tell the proportions of the bacteria that are present based on how much DNA from each species of bacteria we find. Some we see a lot, some a little bit. With culture, we don't have that. With culture, it grows or it doesn't grow, and we can't really get a sense of how much. With this method, we can say how much of each species is present, and so we get proportions, or what we refer to as the relative abundance. It's the term we use. Now let me stop and just go through some quick definitions, because you may hear these words used. You may see these written in some literature about the CF. So we talk about the microbiome. I don't know how that translates into Italian, but it's a microbiome. The microbiome we refer to as all the microorganisms, the bacteria, the viruses, the fungi, and their genes. Microbiota is what we call the microorganisms residing in a distinct place. And this is, as I referred to earlier, as the microbial community. So the microbiota means the microbial community. When we talk about the CF microbiome, we're mainly talking about the bacteria that are present in the airways. Diversity is a concept that I want to introduce to you because it becomes important when we start to think about some of the things we're finding with the CF microbiome. The diversity refers to the number of different bacteria that we find in a community and their proportions in the community. Both measures together, what is their proportion, relative proportion, that is a measure of what we call diversity. And let me show diversity also this way, because it's an important concept. Again, it's the number of different members in the community and the proportions of those members in the community. I'll give two examples here. We have these pie charts, and each color is a different, say, species, a different member of the community. So two communities, A and B. Each of these two communities contains 6 members. You can see each has 6, but you can see that they're in very different proportions. In community A, they look pretty even. Community B, you can see that about half of that community is just 1. We can also look at this way, where you can see the proportions. Community A looks pretty even. Things are proportionate. Community B, half of those are made up of the species in the blue color. And so A has more even proportion, and we would say that A is more diverse than B. So diversity is an important measure. So now, what have we found using these approaches? Well, one of the studies we've done fairly recently is we looked at a large number of samples from people with CF, 945 samples. We did both routine culture in the laboratory on the plates, and we also at the same time did the DNA analysis that I'm talking about. And what we find is that about half of the DNA did not belong to any of the bacteria that were grown in culture. In other words, we're missing a lot, as we suspected, of things that were present but that we weren't finding in the cultures. And in fact, that half of that DNA represented many, many, many more species than the few that we were finding in culture. So a typical culture would say maybe there's 1 or 2 things there, maybe Pseudomonas and staph, maybe Pseudomonas and Chromobacter, or one of these other things that we hear about from time to time. But when we do the sequencing, we find dozens of different bacteria present. So again, the culture is showing us the tip of the iceberg. There's a lot beneath the surface. So let me discuss some of the early findings we've seen with some of these approaches. One is we're very interested in this change in diversity with age. The thinking used to be, and I think some people may still think this way, that as people with CF age, they get more and more and more different types of bacteria in their lungs. We would expected to see that diversity was increasing with age and with lung disease. And in some ways that seems to make sense when we think about it, because we have children who first had staph, and then they had staph and Pseudomonas, and then maybe they get Pseudomonas plus Aspergillus, or plus mycobacteria. So the sense is that they keep getting more and more and more. We were very surprised. We saw the opposite, in fact. It's not true. This is a study now that looks at different age groups. And you can see the age groups along the bottom. And diversity is on the other axis. And each point is one sputum culture, one sputum sample. And the line, I don't have a pointer, but the line in the middle is the average. And you can see that the average diversity goes down with age. Down and down and down until the 30s. And then it goes up a little bit at the end. And we have an explanation for that, but just for now, the important message is diversity actually drops, it doesn't go up. We believe that a large part of diversity going down is both, it may be membership, maybe there are fewer things, but it also is that the communities become less even. So one thing starts to take over, and it crowds everything else out. And as I showed you in the previous slide, when there's a less even distribution, we say that the community has decreased diversity. More evenness is more diversity, less evenness, less diversity. So diversity is going down. What have we learned about early microbiome in infants? Well, it used to be thought that infants with CF, like all infants, have very, very little bacteria in their lungs. And again, the bacteria accumulate with age. Well, it turns out that infants actually have fairly high levels of bacteria in their lungs very early on, much earlier than we thought, with much higher levels of bacteria than we previously thought. But at a fairly early age, diversity already is starting to decrease. Earlier than we thought. We thought initially that that decrease in diversity happened in the second decade, maybe towards the end of the first decade. It turns out, within the first few months, you start to already decrease the diversity. And as one of the, what we would call traditional bacteria in CF, Pseudomonas, staph, as one of those bacteria gets in the lung, then we start to see diversity already starting to go down. The feeling is that one of those traditional bacteria starts to crowd out everything else. It starts to take over. And as that happens, we see also at the same time increasing inflammation in the lung. So this is telling us now, using these methods, it shows us how much more we can learn than what we were able to learn with just culture, because now we're understanding a little bit better what happens in early lung disease. And I think this is giving us important insights that may, we believe, translate into better approaches to therapy. Here's another study fairly recently that tries to associate the types of bacteria, the community, with lung disease. So this is a group from Dartmouth in New Hampshire, looked at they sampled different areas of lung with a scope. They went in and washed different areas, different lobes of lung, and what they found was different communities of bacteria residing in different parts of lung. And they also realized that those different parts of lung had different levels of disease. And we've known this for quite some time, that lung disease in cystic fibrosis is not always even across the entire lung. Different areas have more or less disease. And what this group showed was that those different areas of disease had very different looking communities. Now, what we don't understand, though, this is one of the difficult things to try to sort out, is does the different levels of lung disease cause different communities to be there? Or do the different communities of bacteria cause the lung disease? Which is causing which? We have an association, we don't know the cause, which direction things go. And it may be a little bit of both. But again, it's teaching us that these different communities of bacteria are important. They seem to matter. There are correlations with disease in a variety of ways. This is just one example. So finding these different communities isn't merely an exercise of, isn't that interesting? See what we can find. We're really starting to piece together these different types of communities with different degrees of lung damage. And I think this is going to draw a very important picture. Again, things that we would have never been able to learn just with culture. Now, let me talk a little bit about pulmonary exacerbations, something that we're very interested in my lab in trying to understand. There's lots of other laboratories are trying to understand. You know that these are flares of respiratory symptoms. They're generally unpredictable. We don't know when they occur or when they're going to occur, but they are associated with a decrease in lung function. And so these are events that we would like to prevent and that we would like to be able to treat better. But we don't know what causes them. We believe infection is part of exacerbation because we treat these with antibiotics and most people get better. So we believe there's an infectious part, but it's really not entirely clear what causes exacerbation. The antibiotics that we use typically target the main bacteria that the lab tells us is there, Pseudomonas or Staph burkholderia. Again, what causes? Well, for a long time we thought all sorts of theories about infection and exacerbation. People thought, well, maybe exacerbation means you get a new bacteria. If you don't have a Pseudomonas and now you get Pseudomonas, maybe that causes exacerbation. Some people thought, well, maybe exacerbation was that just the total amount of bacteria, the Pseudomonas, just increases in the amount that's there and causes exacerbation. So we have to treat it with antibiotics to decrease it. But we really, again, these seemed like logical explanations, but we didn't have any good proof of this. Well, what we find with the approaches that I'm talking about is that, in fact, there is no change in the total amount of bacteria that is there with exacerbation. We don't find that a new bacteria occurs at the time of exacerbation. And very interestingly, and completely opposite of what we believed, we often see that Pseudomonas decreases at the time of exacerbation. So you can see on this chart, the things that are increasing are to your right, things that are decreasing go to the left, and you can see Pseudomonas actually decreases. Some of the bacteria that the laboratory would not culture and that we find with DNA are the ones that are increasing. So it's giving us a very different sense of what we believed for many, many years about what is going on with exacerbation. It's dispelling some old myths, and it's telling us what we thought about Pseudomonas, for example, doesn't seem to be correct, which raises interesting questions about treatment. Should we be going after the Pseudomonas when we have an exacerbation, or should we be using an antibiotic that treats something else? Would that be a better approach? Well, we don't know at this point, but we're finding some very interesting things. Let me just mention, we're doing a study currently to try to understand this a little bit better that involves collecting sputum samples from patients daily for years. And the reason we're doing that is we want to collect samples leading up to exacerbation. Because exacerbations are unpredictable, we have to get the samples right before that happens. The only way to do this is to get every day, have very, very large collections, and we can analyze the samples in that period that person is moving from baseline to exacerbation. Now, another thing that we're very interested in and is a very difficult thing to study are interactions between bacteria. So we've explained that there are many, many things present, not just the 3 or 4 things that we traditionally think about. There are dozens of different species present. And what we know is that when bacteria are in the same location, they interact with each other. It's almost, it would be very unusual, I believe, for 2 bacteria to be in the same place and not be interacting one with another. There's a number of different ways bacteria interact. They can compete with each other. So one species may suppress, antagonize, we would say, another species. Sometimes there's cooperation. They like to be next to each other. They do better when they're next to each other. Sometimes one will protect another species. So one species of bacteria may make a product that destroys the antibiotic that might be effective against the other. So there's these interactions that are positive. We also know that bacteria have a way of communicating with each other. This is called quorum sensing. A lot of studies about this lately, but we're recognizing the different molecules that bacteria produce that allow them to talk to each other. Many, many other potential interactions that bacteria have. And so we're very interested in trying to understand when we have these large collections of bacteria, in one place, in this case in the airway. How are they interacting? Are there some communities that are more dangerous than other communities because of the interactions between bacteria? So we are making a lot of progress in understanding how these communities are organized. The theory is that these communities of bacteria are not just accidentally formed, that there's a pattern, that there is rules that govern how bacteria associate one with another. This becomes very, very important if we want to think about ways that we might be able to influence how those communities look and force a community into a place that may look a little bit better or may be less bad for the lung. One of the ways we do this is what we call network analysis. And this is just a depiction, a diagram showing the ways that these different species of bacteria can interact with each other and how they associate. And the red lines mean that there's a negative correlation. We hardly ever see these bacteria associating. The other lines show the bacteria often go together. Right? So now we're, again, trying to, in a more sophisticated way, understand the rules of how bacteria form these groups. Not all groups are the same, and we believe some groups, again, might be, I won't say healthy, but better than other groups that may cause more disease. Again, important to understand that, because if we understand that, then maybe we can help move things from one form of community to another form that is less dangerous. That's the hope. Now, you know a lot about CFTR modulators. These are the new drugs that there's a lot of excitement about. A very strong pipeline of development. You can see here drugs that are in development, those that have already been approved for use in CF. This is a very, very robust development pipeline. Many more companies are now developing these drugs. This is just a partial list. There are many more. So we believe that in the next couple, 2, 3, 4, 5 years, there'll be many more of these for children with CF with all sorts of mutations, not just the most common ones. And these are, I think, going to really be game changers. They're going to really very dramatically change CF. They're going to change life expectancy. What we need to understand is how these impact infection. So we know from early studies now that with the first one that was approved, Kalydeco, this reduced the rate of Pseudomonas infection in people with the mutation, G551D mutation, that Kalydeco works best for. 30% of people with Pseudomonas became culture negative 1 year after starting Kalydeco. So we thought this was fantastic. Not only is the lung doing better, it seems better able to kick out the Pseudomonas. Well, in fact, what we found now with more study is that it's not quite as good as that. We see that even though it rapidly reduces the amount of Pseudomonas, the Pseudomonas gradually increases again over time. And you can see in this graph here, the green line this is in months, and decreasing Pseudomonas, but then gradually over several more months, the Pseudomonas starts to creep back up again. And we're not sure really why that is. We need to study this more. We don't know yet the impact of the other new drugs, or Cambi or Symdeko, but we're doing studies along these lines to understand this. New therapies in CF, many antibiotics in the pipeline. This is just, again, a small list. I can tell you there are dozens and dozens of new drugs being developed that are antibiotics intended for use in CF. Many, many of these. And similarly, we don't know what these drugs are going to do. Some of them are very broad. They kill many, many bacteria. Others are very narrow, very selective, kill only one type of bacteria. But we don't really know how these will affect that community. In the past, as I mentioned, we were only interested in studying what a drug did against Pseudomonas. And that's really all that the regulatory agencies, the ones that approve antibiotics, were interested in. Just Pseudomonas alone. But now we have to understand a little bit more. What about eradication therapy? You know that this is widely used for Pseudomonas, so the first time someone is infected, we treat very aggressively. It's not standard for staph, it's often used. The effect of this eradication is not clear for other bacteria. There we go. The questions, how does this approach affect the entire community? We don't know. Is it possible that with this early aggressive therapy we can decrease the total amount of bacteria, or if we eliminate some species, will others just come in and take their place? We don't know. This is something we're, of course, very interested in studying. So what have we learned? Let me summarize what we've learned. We've learned with this new approach that there are these complex communities. The bacteria that are not cultured often comprise the significant proportion of the community. We know that bacteria interact in many, many ways that likely affect disease. We don't know nearly enough yet about how those interactions work and how they affect disease. We know that communities differ between individuals with CF, but there are some common themes among these. As I mentioned, surprisingly, we found that diversity decreases with increasing age and lung disease. This is mainly due to decreased evenness as one species begins to dominate the community. But the total density stays about the same over time. The total amount doesn't go down. Questions remaining. Well, no single bacterial community feature explains the differences that we see between people. We have not yet figured that part out. I think we're making good progress. No single change predicts exacerbation, although we have some hints, as I mentioned. Again, not clear how these interactions impact health. As I mentioned, impact of the new treatments, eradication therapies, the modulators, new antibiotics, largely unknown, will need to be studied. But the good news, despite all these questions, is we have developed very good tools now to take a new approach and to understand all these things. So, is the glass half full or half empty? We've learned a lot. It's complex. There's a lot more we need to learn, but we're making good progress. I think we're going to learn an awful lot more in the next several years, and it's really going to change our approach to treatment of infection in CF. I think it's a very, very optimistic time. My funding is from the Cystic Fibrosis Foundation, the National Institutes of Health. Grazie mille.
Thank you very much, John. There's time for questions. One consideration from my side, I speak in English so that it can be translated and you understand, that we understood that the situation is much more complicated than we thought. And I was a little bit hesitant about inviting John here because essentially we understand that we have been thinking of a system which is relatively simple to fight, one bacteria at a time. And now we understand that the system is much more complicated. And this term of diversity, which comes out now, is a new term that we have to think about and what to do with it. And I thought that it was good anyway to invite him and to give an opportunity to give this talk, because then we learn that, first of all, there are many, many things that are going on that we can understand. And second, we understand also that there are good hopes for totally new ways of approaching the problem. So this is not a question, it's just a commentary, if you like. Maybe the question is, if you put yourself 10 years from now, and you think about what could come of good out of this kind of approaches, what you would envision. Huh?
Well, the hope is that as we learn more about the complexity, and if it's true that there are some communities of bacteria that are associated with better health, and we can better understand how these interactions between the bacteria occur, we can do things that force the community into a direction that is more favorable than a bad community that seems to be associated with a lot of disease. Now, the best would be if we had no bacteria in lung. We had incredible antibiotics. If the CFTR modulators got rid of all that would be the home run, to use a baseball. That would be the big win. Maybe, but if we can't get quite to there, or I should say before we get to there, I think we will get there, but before we get there, if we can have this understanding where we can modify our therapies in a smarter way. Because in many ways right now, the therapies that we use is a big hammer, just across the board. And maybe that's, I think, as I said, it's good because we saw that really good aggressive use of antibiotics in the 1960s increased life expectancy. So I would never suggest that antibiotics are not good, but I think there are probably smarter ways to use antibiotics. And the only way that we can use antibiotics in a smarter way is to understand some of these principles. So that's my hope.
Think about the question. I can translate from Italian to English. So.
First of all, thank you for this very nice and clear talk.
Thank you. Thank you.
I want to ask you, in recent years there is a great interest for microbiome, also gastrointestinal microbiome. And it is supposed that there is a relationship between airway and gastrointestinal microbiome. We participated in Milan in a trial funded by the Cystic Fibrosis Foundation on the use of probiotics. It was a placebo-controlled trial, multi-center trial, but unfortunately we did not we could not confirm earlier finding of smaller studies with monocentric study, and we did not find any effect on the number of exacerbation or in other FEV1 and body weight. So I agree with you that it would be very nice to have other approach to to treat lung disease and the infective pattern. Do you have experience with probiotics on the airway microbiome, and what do you think? It would be a way that we should pursue further, or it's a closed?
Well, my only experience with probiotics in gut is your study, which was one of the best that's been done. So I'm familiar with your study with the lactobacillus. I think there's a very strong rationale for doing this. We need to, again, better understand this relationship. There's a lot of new information, experimental and observational studies that show that there certainly is a link between gut and lung inflammation. We also know that the gut bacteria in CF are very disordered for a number of reasons. The CFTR is not only in our lung, but it's in our gut. And so that causes a different gut community. All the antibiotics we give people with CF disturb the gut microbiota. So there's many reasons why things are disordered in the gut. And there's increasing evidence that, as I mentioned, there is this signaling of inflammatory mediators between gut and lung. So I'm hopeful that despite the results of the study, that there will be more funding in this area, because I think it is worthwhile to pursue. I'm often asked this question, could I imagine a probiotic sort of approach directly into lung? Well, that sounds a little crazy, because we, for 60 years, have been trying to get rid of bacteria in lung, and now should we suggest putting bacteria into lung? I would say, and whenever I'm asked that question, I say, well, I think that the probiotic approach is going to be in gut that's going to affect lung. Yes. So I still believe this is going to happen. I also think that you could also think about a probiotic effect as a more selective antibiotic effect. If we don't use a big sledgehammer and we just use a little targeted, could we shape the microbiome? So in a way, it's a probiotic approach, in a sense.
So do you think that different probiotics should be looked
it may
for?
be. I think we need to understand some of the mechanisms. I think that your trial was a shot in the in the right direction, but was that the right strain to use? Was it the right dose? Was it, who knows? There are many, and I think we could continue to do those types of studies and hope that we find a good result. Or maybe we say we need to understand the mechanisms a little bit better. We need to understand a little bit more about how this signaling happens so that we can design the trial in a better way. It sounds easy to do. It's not easy to do. But no, but I believe there is a future for this. I really do.
Thank you.
I noticed that you never spoke about the gene mutation in CF, except in the case of the ivacaftor. So since there was a big debate about the relationship between genotype and phenotype in CF, is it still update this debate? I mean, are the lung infections still affected by the genotype in cystic fibrosis, or this is something not yet updated?
Yeah, it's a very good question. I believe that there will likely be correlations between genotype and the types of bacteria that we see, the different types of community. We've not studied that in a serious way yet. In order to answer that question, we would need to do a larger multi-center study. The study that we've done, most of the work that I've done is really involved only our center. So of course, most of what we have are delta F508 mutation. We would need to have a much larger study that involves the less common, which would involve many centers. It's a very good question. My expectation would be that it does matter. There is a correlation, just as there is with genotype phenotype. We would consider in some ways the microbiota as a phenotype in a way. And so I would expect that we would find that. Yeah, it's a good question.
Buongiorno, mi chiamo Francesco Del Bosco. Io devo dire che sono rimasto un po' sorpreso da questa relazione, nel senso che io sono un malato, quindi, se mi permette la battuta, credo che sarebbe auspicabile che la ricerca la facessero i malati, cioè bisognerebbe che i malati si mettessero a fare un po' di ricerca, perché forse arriverebbero prima al punto, nel senso che quello che ho sentito è molto interessante dal punto di vista così della letteratura, ma personalmente credo che andare alla ricerca di tutti i possibili antibiotici, di tutte le possibili interazioni che possono avere i vari batteri che colpiscono una persona malata sia un po' come cercare un agono in pagliaio. Una cosa che mi ha molto colpito è che mentre in alcuni all'estero, soprattutto in America, in Inghilterra, in Belgio, sta venendo fuori come uno dei problemi più importanti della fibrosi cistica, che è un vecchissimo problema, sia questa incapacità di gestire gli acidi grassi, cioè di non essere capaci di metabolizzare gli acidi grassi, e che questo dismetabolismo degli acidi grassi impatta profondamente sul sistema immunitario. Allora, tutti questi batteri, da quello che io so, mi corregga professore se sbaglio, sono ospiti nel nostro organismo, cioè noi li abbiamo naturalmente nel nostro organismo. Per qualche ragione il nostro sistema immunitario a un certo punto impazzisce, non riesce più a tenerli sotto controllo e cominciano a dare i problemi. Ma quando io ho letto che che i malati di fibrosi cistica non sono in grado di metabolizzare l'acido arachidonico e che l'acido arachidonico innesca una produzione metabolitica come le prostaglandine, le leucotrieni, i trombossani, e che questi a loro volta inducono la produzione di interleuchine come l'interleuchina 4 o 13, che vanno a colpire le cellule mucosali, quelle che producono il muco, e quindi favoriscono la produzione di muco. Allora, se l'aumento del muco è il substrato necessario e indispensabile per produrre questi batteri, non sarebbe meglio cercare la strada che riduce la produzione di questo muco, quindi facendo attenzione su quali alimenti i malati di fibrosi cistica mangiano, su quali sostanze sono in grado di scatenare questa produzione di muco? Perché noi dobbiamo essere in balia delle interleuchine 14 quando esistono delle sostanze, come ad esempio gli omega 3, che sono in grado di contrastare? C'è uno studio belga molto bello che è stato fatto qualche anno fa, commissionato dall'Associazione Fibrosistica belga, in cui hanno somministrato gli omega 3 ai malati fibrosistici e hanno visto che le esacerbazioni infiammatorie diminuivano e che quindi era necessaria una terapia antibiotica minore, con una migliore della qualità della vita. Io, ecco, di fronte a tutto questo sono sconcertato. Non è possibile che oggi non si tenga conto del dismetabolismo degli acidi grassi nelle persone che hanno la fibrosi cistica, quando l'imbalance tra omega 6 e omega 3 è alla base della produzione di metaboliti che innescano un'infiammazione sempre più grave e sempre più profonda e sempre più preoccupante. Ma qual è la strada? Noi andiamo dietro a sempre maggiori infiammazioni, con sempre maggiori farmaci, con sempre maggiori...
At the beginning, the question was about the use of let's say the importance of acid fats in the immunitary system, and whether this should be more important aspect to be considered rather than the kind of study that you have been.
Yes. Well, thank you for the comment. I think this is in line with the question about probiotic and the manipulation of the gut to influence lung. There is a story about omega fatty acids, as you mentioned. It's not an area that we've pursued actively yet, but we will be approaching that to some degree by looking, I didn't mention in the talk, in addition though to looking at just the bacteria that are present, we'll be looking at their metabolites. So we will be doing a metabolomic analysis of the bacteria that are in the airway. Some early work in that area has shown that fatty acid, and particularly short-chain fatty acid, is very important in inflammation, very similar to what we find with gut. So my area of expertise and my interest is, again, as a microbiologist, is focusing on the bacteria, but I think there's going to be a very important story to tell about gut host metabolism, bacterial metabolism in gut as well. I don't know where the omega story stands at the moment, but I think that that story is going to continue. It's not done. It's not an area, though, that I know a lot more about. But I take your point, and I think it is another area that should be actively investigated.
Posso aggiungere anch'io qualcosa? Abbiamo eseguito un trial multicentrico italiano anche sulla supplementazione con DHA in pazienti con fibrosi cistica, e anche qui noi abbiamo trovato degli effetti rilevanti sulle riacutizzazioni respiratorie, sulle prove di funzione respiratoria, sullo stato nutrizionale. Il nostro studio è entrato in una meta-analisi, una Cochrane review. Il gruppo Cochrane della fibrosi cistica si occupa di analizzare studi sullo stesso argomento, di metterli insieme, a volte interpellando i vari centri che li hanno condotti, mettere insieme tutti i dati in modo da avere numeri molto elevati, e purtroppo la Cochrane Review, abbastanza di pochi anni fa, ma queste vengono aggiornate periodicamente, perché se escono nuovi lavori viene rivisto il tutto, ha concluso che al momento non c'è alcuna evidenza di efficacia sulla supplementazione con DHA certo che il deficit di omega 3 va corretto, quindi i nostri dietisti stanno molto attenti che vengano ingeriti, i grassi ingeriti abbiano una composizione ottimale e che includano anche gli acidi grassi essenziali, in particolare gli omega 3.
Allora, il trial, mi perdoni, ma non è necessariamente una garanzia, nel senso che esistono dei trial che dimostrano che gli omega 3 fanno bene al cuore e ci sono dei trial che dicono il contrario. Quindi bisogna vedere come è fatto il trial. Allora, io le posso citare, se vuoi glielo do, uno studio fatto da questo Thyssen e Thibault, due ricercatori belgi, che hanno detto che invece...
Sì, ma sono entrati nella meta-analisi.
Ecco, ma attenzione, omega 3, attenzione, dare degli omega 3 e poi non andare a misurare nel sangue se ci sono. Il vostro trial li ha misurati nel sangue, cioè man mano che davate...
Certo!
E questi aumentavano?
Sì, aumentavano, ma non c'era un effetto sulla...
Ma arrivavano valori normali? Lei sa che il rapporto dell'imbalance è dato, come dire, ottimale tra 4:1. Cioè, se io do gli omega 3, ma li do in una forma che non possono essere biodisponibili, oggi c'è un grande dibattito sulla biodisponibilità degli omega 3. Ad esempio, ci sono, mi permetta, ci sono omega 3 che vengono dati in forma trans quando il nostro organismo non è in grado di assimilarli. Allora fare un trial, dando come ha fatto l'Istituto Mario Negri di Milano sulle materie cardiovascolari, e ha dato degli esteri etilici che sono i più scadenti che esistano, e dandoli quasi...
Noi non abbiamo fatto questo.
Allora, però li avete misurati, avete visto se questi aumentavano nel sangue, se arrivavano a valori che potevano essere riportati a valori normali? L'avete fatto questo?
Ma certo, lo studio ha preso in considerazione...
Avete fatto il profilo lipidomico per vedere se questi acidi grassi che voi andavate a correggere...
Lo studio di qualche anno fa, e comunque, le ripeto, non è un capitolo chiuso. C'è chi sta ancora indagando, ci sono tanti preparati che sono diventati oggi disponibili.
No, ma io trovo sconcertante che oggi la ricerca sulla fibrosi cistica non tenga conto delle ultime ricerche.
Ma non è vero che non tiene conto.
Voi non sapete che cosa siano le resolvine, le protettine, le maresin, prodotti che derivano dal DHA. E che queste sostanze sono carenti nelle persone che hanno la fibrosi cistica. Il loro problema è che loro ogni volta che mangiano qualcosa assumano acido arachidonico e innescano dei meccanismi infiammatori. E voi che cosa fate? Li curate con antibiotici. anziché ridurre le esacerbazioni infiammatorie. Gli omega 3 hanno la capacità di... Questo studio di Thyssen e Thibault dimostra, loro dicono molto dicono ci sono studi, non è ancora definitivo, bisogna fare altri studi, altri trial, ma noi dando gli omega 3 abbiamo ridotto le esacerbazioni infiammatorie alle persone. Poi non basta dare gli omega 3, bisogna vedere se uno si prende gli omega 3 e poi va fuori a farsi una pizza e si mangia o le patatine fritte e introduce acido arachidonico, i suoi omega 3 vanno a farsi benedire. Allora, Sig Miller in America sta facendo un lavoro grandissimo sull'alimentazione da fare sulle persone con cistica.
È importantissimo quello.
Ecco, ma non c'è traccia. Voi non parlate mai di acido arachidonico nei vostri lavori.
Non è vero, scusi, non è vero. L'anno prossimo parleremo di questo, ma non è così.
Ho i dati del 14° congresso della fibrosi cistica fatto a Verona. Non c'è una riga sugli omega 3, non c'è una riga
qual è il 14° congresso?
Quello sul congresso di primavera tenuto a Verona nel 2016, che fa il punto della ricerca.
Lì deve chiedere, si vede, i ricercatori italiani non avranno presentato un progetto del genere. Noi l'avevamo presentato a suo tempo.
Scusate, sarà la discussione...
Sì, la rimaniamo, magari ne parliamo poi.
Ci sono altre domande? Volevo fare un ultimo commento personale, magari se lo volete tradurre. Il professor Lipuma, tra le varie cose, ha una banca di coloni che comprende...
Use a microphone.
So, a final consideration, Professor Lipuma has a bank of 35,000 sputum cultures. So it means, and he has been looking at the sputum culture every day for a lot of patients. And then this is just one of his studies. So, I mean, the story of how we go against cystic fibrosis is very complicated. There are a lot of things to be done. If you think about it in the course of history, we have been doing things, thinking about the famous, let's say, genetic therapy that never came to give good results. There might be other routes to follow. The problem is that if we don't follow all the routes, we will never be able to find the right one. So I think we have to, of course, follow also the one which has to do with understanding the structure of the microbiome and the way in which the bacteria compete with each other. It's complicated, but if we don't do it, it'll be very hard to succeed. So I would like to thank you again, Professor Lipuma, for being here. And I don't know,
Grazie. Grazie mille.